Good afternoon, everyone. We'll start our session now. I'm Cal present. Let me know if it is visible or not. So, is the screen visible to you all?
Yes, visible.
Okay, nice. So, we are in the week 8 of the problem solving session, and this will be basically focused upon the content that were there in week 8 of the course. So we will discuss few questions and so let us start that. So this is the question first. Which of the following is an online repository of the targeted proteomic assays for detection, validation, and ultimately quantification of the proteomic samples? A. Pride, B. PESEL, C. Proteome Exchange, and D. SRM Atlas. So, amongst these four, which is used for quantification of proteomic samples? Any guesses? So, anyone wants to try? What will be the correct option? So, it's an online repository for targeted proteomic assays. So, where you can deposit your targeted proteome assays so that it will be publicly available to the entire scientists and people working in the field of proteomics. So, suppose anyone wants to do let's say a targeted proteomic study for protein X, and you have already uh, made the method and everything for a particular protein. Okay, you have let's say you have done an MRM study or you have done an SRM study, and you have all the information about the target ions, reference ions, transitions, which all transitions, and what is the collision energy, etc., etc. All the information. Then you can deposit that in these different online repositories. And suppose, let's say, there is some another person who also wants to work on that protein X which you have already worked upon, and he wants to perform an targeted study. So then he does not have to start from the scratch. He or she can use the information that you have already deposited in the online databases. So there are different kinds of databases. So amongst these four, which one is for quantification? Is anyone speaking? I'm not able to hear. Hello. So basically, if we see, so Pride is a proteomic identification database. Let's see. So I think anyone still wants to try, or I should discuss. Okay, then I'll discuss this. So,
Yes, ma'am. Please discuss.
so okay. So Pride is a proteomic identification database. It is basically managed by European Bioinformatics Institute. So this is a database where you will have, well, you can submit MS based proteomics data set. Okay. So it will have all kind of proteomic database. Okay. Now PESTLE is Peptide Atlas SRM Experimental Library. So here you will have specifically a library of SRM experiments. So SRM is one of the targeted proteome approach, and here the library will have specifically for SRM experimental data. So if you want to, uh, so what is the question? So yeah, so if you want to, let's say you are trying to do an SRM study, targeted SRM study using, let's say a protein, whatever is your protein of interest, protein X, then you will search using this particular library whether first you have to see whether your protein of interest is present in the library. Second, the instrument. So there are different mass spec available. There are different companies like Agilent, Ciex, Thermo, and every instrument, depending upon the company, there will be slight different in the parameters. The principle, the mass spectrometry principle will definitely remain the same. However, there will be slightly different parameters customized depending upon the different companies. So that is why you have to also check for the company, and whether the kind of instrument, like whether it is cute of mass spec, or it is triple quadrupole, or it is ion trap or orbit trap. So different kind of mass spec will have a different principle, as well as the company, because there will be difference in the parameters as well. Then coming to proteome exchange, so it is a consortium set up for the coordinated submission and distribution of MS-based proteomics data across different databases. So PRIDE and this Peptide Atlas, these are different databases where people submit their data. Okay, and proteome exchange is a big consortium, is a association kind of, which looks after these different submissions. They have also published different guidelines for submission, how uh, if you want to submit your any experimental data in this different databases. So it is a big organization, and you can get the data across different databases using Proteome Exchange. So it's a association which looks after different kinds of databases and all. And the fourth option is SRM Atlas. So here it is a collection of targeted proteomic assay for detection and quantification of proteins in complex proteome by mass spec. Okay. So the correct option is option 4. So if you see what was asked, it was asked which of the following is an online repository of targeted proteomic assays for detection, validation, and ultimately quantification of the proteomic samples. So SRM Atlas is where you can have a collection of targeted proteomic assay for detection and quantification of proteins. So the correct option is this, option 4. Now, going to question 2. So, here this is a small exercise kind of thing. So, what I will do, I will share this. This is a FASTA sequence of a particular protein X. Here it is mentioned. So, from this, the sequence is given, and there are different tools, online tools available like BLAST. So from that you have to find out certain things of this particular sequence. So what is mentioned, Rashi is working on a breast cancer. Okay, she wants to validate one protein X using targeted proteomics, and the FASTA sequence is given below. Okay, so this is the objective of our Rashi, and here is this question second. So find out the name of the protein next using BLAST. So I will give you the link in the chat box as well as this FASTA sequence. So it's a protein sequence, and from that you have to find out what is the protein. Okay. Now, so I'll just stop sharing for a while. So I have shared the link for BLAST, and then I'll share the FASTA sequence. So I have shared both the FASTA sequence as well as uh, the BLAST search. So what you can do, first go to the BLAST tool and then give this FASTA sequence there and try to find out which kind of protein it is. So at least open this BLAST search tool. Everyone has opened the BLAST search tool?
Yes, ma'am. It's open.
Okay, nice.
Ma'am, after that,
Yeah, after that, so even here I will show you. I'll share my screen again. So something like this would have opened if you have opened your that link blast, right?
yes, ma'am.
Okay. So here, this is the section. So here you have to give that FASTA sequence or if you have some accession number. So basically, BLAST is used to find. Suppose when you are working in a proteomics or genomics, when you don't know what is your protein of interest. Let's say you found some new protein, okay, and you don't know which protein it is. So what you can do, this is a database. From here you can get the information about what protein it is. Okay, so I will paste here my FASTA sequence. So this is the FASTA sequence. So make sure there are no gaps when you are giving this FASTA sequence, otherwise error will come. So either you can give the FASTA sequence here, or you can also upload a file if you have in some other format. So you can also upload the file of your FASTA sequence. So there are different kinds of blast search you can perform. This we are performing for protein search. So you can also perform blast N, which is for nucleotide search. So if you want to find out DNA sequence, if you have a DNA sequence of a particular uh, protein or anything and you want to see what it encodes for, you can even do that. Then you can also, even from the protein sequence, you can find the DNA, which is the DNA of it, and even from the DNA sequence you can find the protein. So this is if you can see here, this here blast X is search for protein database using translated nucleotide query. This is search translated nucleotide database using protein query. So here you can give your protein sequence and then it will give you an idea of a tentative DNA sequence, and from that what is the DNA tentative, how your protein sequence is matching a particular DNA. So this is optional. Here you can also give so I'm giving Homo sapiens. Okay. And we have to perform blast P, protein blast, and then click on this button. So I think there is some error here. Let me I'll repaste this sequence, if because even if there is a slight error in this FASTA sequence, then you will not get the result. So you have to be very sure while pasting your FASTA sequence. Anyone of you have completed the search?
Ma'am, actually, I don't copy the sequence from the chat box.
It's like
Yes, ma'am.
you're not able to copy.
Yes, ma'am.
Past the sequence, it's not able to copy.
Oh, okay. Hmm. Is there any option to share some like Word file or something anyone knows?
Ma'am, will you please send the same on Telegram group so that we can?
Okay. Okay, yeah, I'll do that. I'll share that on the Telegram group. Just give me a minute. I have shared that on the Telegram group. Just check once. So is this sequence available? Have you checked in the Telegram?
No, it's
No, ma'am.
I have shared in that group only. Okay. I'll again try to share if that works. Okay, what we can do, we can go to other question and then we will come back to that question. Because I have shared, if it might take time, I don't know.
okay, ma'am.
Basically, these questions were linked actually. So we will move to, okay, this is also linked to that. But let's just try this. This is slightly different. So here, to move further in her experiment, so Rashi prepared a transition list for targeted experiment. So now she wants to perform a targeted
Ma'am,
experiment.
you don't share your screen.
Oh, okay. So here using SRM Atlas, okay. So different parameters here are already given. Okay, and now she wants to find out the number of transitions she could observe for Agilent triple Q. So she would be having this instrument and she wants to work on this instrument. So there are different instruments also available, and from that you can get the transition list. So this is the link for that. I'll share that and also. So here you have to give some protein accession ID. So let's say we are giving this ID. So P04637 is our ID. P04637. Okay. So from here, she can get an idea about her. So now she knows her protein of interest. So she knows her protein accession ID, right? So she had done a prior search. So that questions are left, but so from blast search, she will come to know the protein, what kind of protein it is, and then she can find out the accession number from Uniprot. Okay, and from that she will get a session number that she will use here, and then let's see different parameters. So you have to select different parameters depending upon your experiment, the instrument that you are working upon, and also these are the databases that you have to select, then labels and all. So yeah, we will just take the parameters as mentioned here. Is brought unique in result, duplicate peptides. So YN BIMs, and then we will do query. And okay, yes. So we have not selected here. So we want to look into the atlas. Matlab the databases we have to see, right? So we need to select what database we want to select. So different options are given. So our protein of interest is of a human protein. So that is why complete human SRM atlas. So SRM atlas currently have different other atlas also, M tuberculosis. So it will have a proteome information of different studies that are done on M tuberculosis, Mycobacterium tuberculosis. Then even it has a data of mouse and yeast. So we will select human SRM atlas. So here you will get this kind of result. So what are all these column represent? So this is the protein. So a session number is given. So you can also click on this show column description, because you will not be able to understand what is pre-AA. This is the sequence, what is follow AA, adjacent SS. So what are these values and what you have to see in this. So what is the question asked? Okay, you have to take this information, and from that you have to find out that how many transitions are possible. Okay, so the question is incomplete. But here she wants to see for Agilent machine what are the transitions possible. So you can see here if you scroll. So these are different instruments. QTOF is a different instrument. QTOF, CE, QTRAP, triple Q. So here she wants to look for triple Q. So we will see here, and let's see. So what you can click here if you don't know what are the information. So pre-AA. So pre-AA, what is that? Pre-AA is preceding N-terminal amino acid. Pol-AA is C-terminal amino acid. This is your sequence. Then this is the suitability score. So we are interested in transition. So you have to see Q1 M by Z, its charge, Q3 M by Z. So Q1 M by Z is the mass to charge ratio at precursor peptide ion. Q3 is the mass to charge ratio at fragment ion, Q3 fragment ion. Okay. So here is the precursor and the fragment ion given, and here the information see if for this particular transition, that is precursor and the fragment ion, precursor is this one and this is the fragment. So precursor and the fragment ion, if the information is available, it will show a spectrum, and if it is not available, it will not show any spectrum. So for this particular protein, if you see how many are available, so you can count based upon this. Okay. So if we see 1, 4, 5, 6, 7, 8. So 16 are available for triple Q. So in a similar way, if you want to look for other other different instruments, even that you can see here, and this is the result. And then if you want to see the transition list, so here, so you can go to these different publications, and these are the files. You can download the files here. So this is the MRM files given. So, and you can get the information about the transitions for your protein of interest from this. You can download the file and then use that data. Okay. So this is how you can find out. You don't have to do the targeted essay from the scratch. Otherwise, you have to find if you have an so for a particular protein, you have to find out the transitions. Then at what particular collision energy it will give the highest intensity. So it's a laborious process. It's a very big process. But here, if someone has already done that, so you can use that information and directly perform a targeted analysis. So you don't have to perform the entire process of finding out the correct collision energy and correct parameters for your experiment. So even the information about the instruments are available. So if it is not available, so if your protein is not available, so if here the result is coming as a no match is found, then you have to perform the experiment from the scratch. Otherwise, you can go ahead using this information which is already available in the database. Okay. So, so coming to next question, Aman is planning a targeted proteomics experiment. Which parameters will be crucial to his experiment? So A, retention time of the peptides, B, gradient of the LC being used, C, dwell time and cycle time of the transitions in the list. So transition, right? Transition is the precursor and the fragmentation pair. It is called the transition. And D is isoelectric point of the proteins in the sample mixture. So what is the correct option?
Option C, dwell time
C.
and transition.
Okay. Any other guess? Someone was saying two. Option two, B.
No, C. I was saying C.
Okay, C. So, whenever you are performing a targeted analysis, of course, dwell time and cycle time is very important for the transitions in the list. You need to provide, you need to provide a particular dwell time so that, and it could, I mean, you have to optimize your dwell time so that your all the transitions are getting appropriate time, so that the data points are collected in the sufficient amount. Now you're using mass spectrometry based and generally LCMS based proteomics. So the gradient is very important. So your proteins, different kinds of protein will have a different chemistry, right? So and you are providing a gradient buffers of whenever you are passing your protein of interest from LC. So whenever you are seeing, I mean, the gradient is very important because it will affect the elution of your protein. So let's say you are performing a targeted experiment, not for one but for maybe more than one proteins, and right. So your protein of interest, how it will elute from the liquid chromatography, it will depend upon its chemistry and the gradient. So there will be some proteins which will elute faster depending upon what buffer you are using, and the the column chemistry, what is the what kind of column it is, what is the stationary phase in that, and how your protein will interact with it. Okay, so even gradient of LC is very important. And third is retention time of the peptides. So as I mentioned, that illusion of the peptides will depend upon their chemistry, right? How hydrophilic it is or how hydrophobic it is. So a different protein will have a different retention time. Sorry, a different peptide will have a different retention times. So even the retention times plays a very important role because uh, so, let's say a particular fragment, if it has more of a hydrophobic groups, and your buffers, your system, your column system is also like more hydrophobic, then so it will interact more with the hydrophobic peptides. So your peptide containing more of the hydrophobic amino acid will be retained for a longer time, and the RT will be more for those particular peptides compared to more polar peptides. So peptides containing more of a polar amino acid. So these all parameters play a role whenever you are performing a targeted proteomic experiment. And the option D is isoelectric point of the protein in the sample mixture. So isoelectric point is important whenever you are performing uh, DIGE experiment, whenever you are doing your two-dimensional separation using SDS page. So once that is done, after that, it does not, isoelectric point of the protein does not play any role whenever you are using mass spec based technology. So because the principle itself is dependent upon mass to charge ratio, it does not have any role of the isoelectric point. So option D is incorrect, and you have to take care of all the three condition whenever you are uh, planning a targeted proteomic experiment.
Ma'am,
Yes,
hello.
yeah, hello.
Ma'am, can you please explain the retention time again?
Okay, so basically, peptides, as I mentioned, so there will be fragments of peptides, and these peptides are made up of amino acid, right? So there will be few amino acids which are more polar, and there will be few amino acids which are more non-polar. So let's say like tryptophan, phenylalanine, these are non-polar amino acids. Now you are using chromatography, right? So if your chromatographic bed is made up of more of the non-polar groups, right? So it will interact, it's like interact with like. So hydrophobic column, the bed of your chromatography is made up of hydrophobic groups, so it will interact more with your peptides containing hydrophobic groups, so it will retain on the column for longer period of time because of the interaction, whereas the peptides which are uh, less non-polar or less hydrophobic, they will elute out first. So so this is dependent upon the characteristic. So different peptides will have different retention time. Okay. So depending
Okay,
upon
ma'am.
the chemistry. So this is all about the chemistry of the chromatography, the chemistry of the peptide. So if you're what kind of functional groups are present in your peptides, the amino acids and all. So depending upon that, some peptides will have uh, they will have a different RT, and other proteins might have different RT depending upon that. So coming to next question, calculate the dwell time for each transition in a targeted experiment. with 350 total transitions, the cycle time is set to 2 seconds. So here the cycle time is given and you have been given transitions, 350 transitions are given. So you have to calculate the dwell time. So what is the dwell time? So can someone do that? It's very simple but very important parameter whenever you are doing mass spec experiments. So be it proteomics or any kind of mass spectrometry based experiments you are doing, the dwell time and the cycle time are very crucial. So has anyone calculated? So basically, dwell time is the time taken by MS to measure one transition. Okay. And cycle time is the time taken by MS for all the translation. So cycle time is basically the complete one cycle the MS will go for scanning all the transitions. So let's say you are giving 10 transition or 100 transition, whatever. So the cycle time is the time taken by MS for all the transitions. So what is given here? So there are 350 transitions, right? And the cycle time is 2. So you can, it's a simple, so you can, just a minute if I can write here. So basically your cycle time, okay, is what 2 second, right? Now cycle time equals to all the time taken by MS for all the transition and and the number of transitions are how many? Number of transitions are 350. So number of transitions equals to 350. Okay. So for 350 transitions, it is taking 2 seconds, right? Because cycle time is the time taken for all the transition, right? So now for one transition, how many? What will be the time taken, right? So it will be simple. Dwell time equals to 2 second by 350. Okay. So which will be approximately 5.7 millisecond. So you can calculate this. So this is very simple. The concept, you just have to remember the concept in this, that what is the cycle time and what is the dwell time. If you remember this, you can easily calculate. So sometimes, these parameters are very important when you are doing a targeted study, as we have discussed. So if the transition, if your cycle dwell time is very less, which means that the MS will get that much less time for a one particular transition. So it will not get, so if your dwell time is, let's say, 2 seconds. So in 2 seconds, it may be possible that the MS is not able to take as many data points as available. So just a minute. So you should always calculate your dwell time and the cycle time. So it should, your dwell time should not be very less also. Otherwise, it will generate very less data points, and you will not get a proper Gaussian peaks for your compounds or peptides. So in this manner, you can calculate your dwell time if the cycle time is given, and vice versa. So generally cycle time is uh, typically little bit more than uh, so if you calculate like 5.7 seconds, or let's say if your dwell time is 5 seconds approximately, and there are 350 transition. So but you should also remember that while going from one transition to next, so ms the instrument will take some time from shifting from one transition to other. Okay, so there will be few seconds of time that will be wasted, right? Whenever the instrument is switching from one transition to other. So cycle time will be cumulative of your transition time plus that extra time required by the instrument for switching from one transition to another transition. So it will not be exact. The cycle time will not be exact of your dwell time into the number of transitions. Going to next question. So Sulekha is doing PTM analysis of the plant tissue sample she has on a mass spectrometer. Which of the following fragmentation technique should be preferred? So, which are the techniques used for PTM analysis, that is post-translational modification analysis? First is collision-induced dissociation, electron transfer dissociation, high-energy collisional dissociation, and option D is collisionally activated dissociation. So this has been discussed in the challenges of proteomics in that week session. Any guesses? So electron transfer dissociation is the technique used when you want to study PTM, post-translational modification. So electron transfer, and there is another technique also, electron capture dissociation. So that is also used for PTM analysis. So this is a direct question. Which of the following is an organization of international significance formed to represent and promote proteomics by collaborated effort throughout the world? It is a scientific organization promoting the development of tools, techniques, and training for the same. So option A is Cancer Proteomic Tumor Analysis Consortium. B is Human Proteome Organization. C is Clinical Proteomic Tumor Analysis Consortium. And option D is Human Proteome Project. So what is the correct option?
B.
It's option B, ma'am.
Yes, correct. Human Proteome Organization. Correct option. So now we'll quickly go through the questions that are left. This one, right? So, so we were here. So I will just directly show if this link works, or we will move to option the next option. So so faster sequence you were not able to see. So here we will go to next question. This is about UniProt.
Ma'am,
So
ma'am,
yes, yes.
ma'am, blast will work if you don't copy the initial or FASTA format. You just need to select the sequence that is starting from methionine.
Okay.
and just paste it in a query box.
Okay, I'll try that. But I think this is required. Let me try that. Um, So have you got the sequence from the Telegram?
No, ma'am, not
Okay.
received yet.
So we'll go to this Uniprot also. Then I'll share this Uniprot link, and I'll also open here. So you can also practice at your own side. Okay. So here you can find your protein. So you can find the accession number of the protein if you have some information. So here from here also you can perform blast. So if you click here, you can either you have to give your some ID or you have to give the sequence in the format, FASTA format. So what is the question from the UniProt? Which of the following is the correct accession number for the protein? So our protein is, let's say our protein is tumor antigen P53. So either you can either you can go to BLAST and give the sequence in the FASTA format, or you can also directly select give the name here. So if the name is cellular cellular tumor antigen P50. So you can just directly click on search. So it will give you all the different the database it has, and it will give you here the numbers. So what is asked? A session number is asked, right? A session number. So these are the accession number. Now our protein of interest is from human. So we'll click on human. So you will directly get here. So this is human. It is mentioned, and P04637 is the first hit coming in the table. So this is the so in this manner you can find out the accession number of the protein. So either you can do this or you can also do using FASTA. We can take this and then paste here. So basically you can give the your sequence here, and here I think there is some problem with the format I have. So there's fair possibility that I have missed some gaps are there. So that is why it is not taking. but here you can directly go to run blast, and from here also you can get the list of the accession numbers. So basically there's some issue with the format, so that is why that error is coming. So you have to be very particular whenever you are pasting the FASTA sequence. So so this is also dependent upon this question. So here, let's say you are giving this protein, cellular, what is the name? Cellular tumor antigen P53. Cellular tumor antigen P53. And we are selecting human, right? So this is the protein of our interest, right? So here you will get the function, here you will get all the information about that protein. And so what is asked, the if you want to see the structure, what is the structure, then yeah. So here you can see different options are given. Function, so here you can see the function, then what are the PTM variants, even that you can see. So glycosylation, phosphorylation, where it is at what particular residue. So all information pertaining to this particular protein will be given here. If you want to see the structure, even that is visible here. So you can also see the structure here. So we'll see. So this is the structure of this protein. You can see this is the structure. So you can select different kinds of things and see what all is available. So if your protein is so when your protein is unknown, all these informations are very uh, important. So uh, when right, so she was working on protein X and she was not knowing what her protein is. She had isolated from particular whatever her experiment was. So this is the procedure that you need to follow. So she's working on a breast cancer cell line, and she had isolated. She wants to validate some protein. So this is how you should perform the steps. So if you have no idea what your protein is, or you are doubtful, then you can use the sequence of your protein, and then you can find out what protein it is matching, and then from that you can also identify the structure, the accession number. So just by so the major hurdle is you need to at least get this FASTA sequence. So you have to first find out the sequence information for your protein, and then you can get all these information. So these are all bioinformatics tools available currently, and you can find out. So this is based upon the function. So you can, as I mentioned, if you click on function, you can see what are the different functions of these proteins, and you can then link how this particular protein, let's say you found out that this protein is getting upregulated in your patient samples and the healthy group, then you can find out that okay, this could be let's say, so P53 tumor, it's a tumor suppressor gene, right? It acts as a tumor suppressor in many tumor types. So there could be a possibility that in case of the cancer, breast cancer, so it would be down regulated. So you will see some kind of change in the pattern of these particular protein different between your control and the diseased condition. So after seeing the functions, then you can identify, okay, this is the role of this protein, then this could be, you can interpret that this could be the possible function, and then you have to look upon further and see the complete signaling, how it is uh, uh, acting, and how it is I mean interacting with other different molecules, and then you can plan your further experiment depending upon that. So yeah, so this is all I wanted to discuss. So apart from that, if you have any doubt, you can ask me. So blast search you can perform directly at Unipriot also and at Blast also, the website. So it's just that there is some issue with I mean the sequence, and that is why it is not giving the result. So even the slight change in the like one, even if the one digit or one number is changed or some spacing is changed, that will not come. So from using this information, you can try to find out the protein of your interest, and whenever you are performing a proteomic experiment. So that's all from my side. If you have any question, you can ask. This is
Ma'am,
the last session. Yes.
I think about the exam this coming Sunday. Do we know what kind of questions we expect? Is it multiple choice or objective short answer?
So it will be mostly multiple choice questions.
Okay,
So I think you will be shared the all these information soon.
okay, okay.
So have you all submitted your last assignment?
For
No,
me,
ma'am.
I have submitted yes.
Okay. So there was one session yesterday also, the live sessions. So I hope you all have attended that and found it interesting. So this is the
Okay, ma'am.
last session. So now I hope you have got the ideas about the what kind of questions there are for proteomics, and you can prepare well for your examinations and assignments. So I wish you all the very best for your examination as well as your future scope for this field if you want to pursue in academia or the scientific field. So thank you very much. If you have any doubt,
Thank you, ma'am.
okay. Okay, so I'm ending the session.